Chapter 15 is all about translation. So now that we have been through DNA synthesis, and you guys know the difference between synthesizing DNA and actually making a mRNA strand of nucleotides, we are now going to need to take that mRNA strand of nucleotides and turn it into a protein for gene expression. So proteins are really important components. They have a billion different components that are different proteins that are involved in various functions in an organism. But the way that proteins are actually expressed or created is by taking DNA, go through the transcription process to make an RNA, and then that RNA is then translated into protein, and it's the protein component that dictates the phenotype. So proteins can come in several different forms. The ones that we're most familiar with is enzymes, but they can also be structural components, hair, bone, communication, transporters. They have various different functions. So proteins are very, very important, and they are coded for in the DNA, so that process is highly regulated. So proteins again are made up of amino acids. You guys have always heard that amino acids are the building blocks of proteins. So there's 20 amino acids, they're listed here in this figure, that make up all the proteins. So you're going to be able to see this figure quite often. So the way to read this type of figure is it has to do with the position of the nucleotide. So the first position is always dictated over here on the left. The second position is dictated up here on the top, and the third position is dictated over here to the left. So if you put all three of those together, like if you have an amino acid codon that has the sequence CCA, then the CCA amino acid is actually proline, abbreviated as PRO. So amino acids are really important because they're going to bind together into a sequence, and this sequence is then going to be folded up into a secondary sequence, very similarly to the double bonded or the double-stranded double helix form, but this again is going to be single-stranded. And then that single-stranded double helix form is then going to be even more folded up into a tertiary structure, and sometimes it's going to be grouped together with other subunits into a quaternary structure. So just keep in mind that amino acids is the genetic code from DNA, and it's going to travel through the messenger RNA strand to be translated into protein. And the amino acid that is going to be created from each DNA strand is called or is coded for by a triplicate code, which we call a codon. So 3 nucleotides for each amino acid is a codon. Codons are, in the whole scheme of things, are actually pretty different. There's only 64 of them. The start codon for every single gene is always methionine, which is AUG. And there's 3 termination sequence or stop codons, and we'll look at those more in depth. And so 64 minus methionine minus the additional 3 gives us 61 sense codons that can be used in different combinations to make up protein or strings of protein. So again, here's that figure again. It looks a little bit different, but the concept is the same. You have your first nucleotide base on the left, the second base is coded for on the top, and the third base is over here on the right. Put those 3 things together and you get some sort of amino acid. So if you count all these up, you'll get a total of 64. 3 of them are going to be your stop proteins, and then your methionine is always going to be your start protein. So when we look at the process of translation, it's basically broke out into 4 major steps, which is a little different than transcription. So translation begins with tRNA charging. So this is where the transfer RNA has to actually be charged and has to bind amino acids in order to start building that amino acid chain. The second step is then going to be initiation, where we have multiple components come together and assemble at the ribosome. And then the third step is elongation, where the amino acids are going to be added one after another to a chain, so just like elongation in any other process. And finally, termination is where protein synthesis is going to halt and the translation components are released. So DNA charging is step number one, and this is where we see a carboxyl group of an amino acid structure actually attached to the hydroxyl group of a carbon atom. And so again, this is going to be taking place on the 3' end of that transfer RNA. So here we've got what our transfer RNA looks like in real life, and it's got a carboxyl group to where the amino acid is going to accept that, or the amino acid is going to be accepted right here. So this component breaks down to an amino acid group, our carboxyl group, which is part of the amino acid, and then the transfer RNA, which may have adenine, cytosine, uracil, or guanine. The second step is actually the translation initiation, and this is where we have multiple components coming together and assembling. So those components are going to include messenger RNA, a small subunit, a large subunit, various initiation factors, and initiating transfer RNA, which we just looked at in the previous slide, and then guanine triphosphate, which is GTP. So once the initiation process has gone through the entire assembly process, the result is going to be a ribosome that is assembled on the messenger RNA strand that has been floating out in the cell from the transcription process. And the first transfer RNA is going to be attached to the initiation codon. The third step in this case is elongation, and this is where we're going to start seeing an amino acid chain being built. So those amino acids are going to be joined to create the polypeptide chains, and the elongation process is going to be aided by many different factors that help with the charge and the delivery of those amino acids. In addition, elongation is where we're going to see the creation of a peptide bond between amino acids, and what we call the P and the A site. We'll talk a little bit more about that in a little bit, and that's going to be catalyzed by peptidyl transferase. So transferase is an enzyme that's going to help with the catalyzation of the elongation process. And finally, the elongation process is going to focus on translocation, or moving down the ribosome to the next position to assemble that next codon. Finally, the fourth step is going to be termination, and this is where the ribosome is going to move to the termination codon. It's going to read the termination codon, and as it reads it, it's going to release factors that bind to the ribosome at this termination point. And the final step of the termination process is that the transfer RNA from the P and the A site is going to disassociate from the original component. So the table that you're looking at right now is straight out of your book. It's table 15.4. This is a really good place to start memorizing all the different components that are associated with this process. Everything that has to do with each of the 4 steps: DNA, tRNA charging, initiation, elongation, and termination.